resisting so that cells survive for a long time actually. Sustaining the proliferation signaling, evading the growth suppressors. So these are the some hallmarks of the cancer, which is important property for any cancer. Then it may be in the metastasis, or it may be localized tumor, or it may go to the detached from the tumor and it go to the other part actually, that is called metastatic tumor. I'm not going in the term, but this is just for the persons who are new to the biology or who are not good in the field of omics actually. This is a whole word of omics. And just think about that. Any human, what they have, organs, tissues, each tissue is made of the cells. You know that cell is the basic unit of any living organism. As we say, atom is the basic unit for any matter. Okay, that's a minimal unit. Same way, any living organism cannot have less than a cell actually. So this is a minimal unit for any living organism. Though there are examples where on the single cell based living organisms are there. Saccharomyces cerevisiae and other examples are there. Okay. Next part is in cell. What is the main part? In cell, there is a nucleus. I'm talking about eukaryotic cell, human cell. In this nucleus, what we have? We have the genetic material. If we sequence the all nucleotides in the nucleus, then this is called whole genome. This is called whole genome. This genome is not just randomly. They are well organized in the form of the chromosomes. Let's say in case of the human, there are 23 pair of chromosomes. And the DNA is well organized in form of the chromosomes. And how they are made? They are made by the histones basically, and this is called chromatins. And there is a modifications of histones is possible. So then epigenomics field is there. When we study the binding of the DNA with the proteins in the chromosome, then we call the epigenomics basically. And the whole DNA sequencing, all the DNA in your nucleus, and in case of the human, it's a 3 into 10 to the power 9 base pairs, because DNA is found in the form of the double helix. So we call base pairs basically. So there is a 2 strand, and each have the 3 into 10 to the power 9 nucleotide. Nucleotide means ATGC. There are 4 chemicals basically. So this study of DNA in the nucleus is called genomics. All part of the genome is not coded in form of the gene. If we study only the genes or mRNAs which are made from the gene, corresponding to the gene, and then this is called transcriptomics. Transcriptomics means all mRNA corresponding to all the genes basically in a cell. So that is called transcriptomics. And this transcriptomics, mRNA converted into the protein, and the study of the all proteins is called proteomics. So this is the definition. There are further definitions are there where there is a protein is glycosylated, then we call glycomics, and if there is a lipid attachment, then it's called lipidomics. Right? Now, question is, that's the basic thing I talked to you. Now I am talking to you that what type of resources we have developed in our group and which you can use for your research. I'm not saying these are the only resources. There are a number of resources developed in this area. I am just presenting the tools developed at our group. Why? Because it's not practically possible for me to cover all the tools. I will give you the reference later on. Okay. So I am focusing today on resources developed from our group. So initial phase, we work on the cancer drug resistance. This is one of the major problems in the case of the cancer, that for some time the drug work, and after some time it doesn't work. The cancer cell become resistant to that drug actually. That is called cancer drug resistance. So I will talk about the two databases developed at our group, Cancer DR and Herceptin-R. Then you know that number of genes are involved in the cancer, and these are called oncogenes basically. What is their properties and what type of information is there? These three databases will help you to understand the oncogenes or cancer genes, genes involved in the cancer. Then next, I will talk to you about the drug delivery. Let's say if you want to deliver the drug or you want to handle the cancer, then how you can handle it. And then I will talk about some cancer biomarkers. Okay. So first thing is cancer DR. So I will tell you here that our group or we are not a traditional scientist. In case of the traditional scientists, they think, they plan, they work in the whole life in particular area and then do something very serious. Our group is basically more sort of opportunist group where we see what type of problems community is facing and how we can help them. Opportunist, why we say opportunist? Because for us, most important part is data. If experimental data is not available, then we bioinformatics can't do anything. So we normally monitor what type of biological data is there in the field so that we can use this data and mine the important information, right? So here, in 2012, we decided that we have to contribute something to in the field of cancer. The question is basically from where we should to start. Then we read in the literature the cancer drug resistance is a big problem. We searched at the time, there was no database on cancer drug resistance. So people are talking in the literature that cancer drug resistance is a major problem, but there was no such type of database out there. So what we did, we collect and compile the information from the literature related to the cancer drug resistance, and we create this database and we are maintaining this database. So anybody working in this field can use this database for their research. Because one thing is there, why the bioinformatics or any informatics based field become popular nowadays, which is not as popular if you go, let's say, 50 years back or 30, 40 years back. Because at that time, experimental data was limited, data was available. If data is not there, what the informatics will do? Nothing. We informaticians are dependent to the data. You got my point? Without the data, we can't do anything. So we got a lot of data, so we create this database, and what this database have? It have the information about 952 cancer cell lines. 952 cancer cell lines on which 148 drugs have been tried and their IC50 value have been checked. And then we calculate what is the target, what is the structure of the target, all type of information, what is the relevant information is required to get it. We get a lot of data from the PubChem, which provide the information about the SH. We get the information from Cosmic and CCAD, where we get the information regarding the genomic profile, like gene expression profile, single nucleotide polymorphism, copy number variations. So all type of information have been collected and compiled in this database. So this is the first database for maintaining the information about the cancer drug resistance. This is the type of information it provides. It provides the structures of all targets and their mutation informations. The complete information is available here. There was another database that's very specific. There is one treatment commonly used to treat the breast cancer patients. That is a monoclonal antibody called Herceptin. Herceptin is a name of monoclonal antibody, monoclonal antibody, which is used to treat the breast cancer patient. Problem with this Herceptin is that it is not suitable to all. You cannot give blindly to everybody. Actually. Okay. In some time it works, sometime it doesn't work. There are some biomarkers already there in the market where they check the HER2 overexpression. If HER2 overexpression is there, then this may work, otherwise it will not work. But this biomarker again have the limitations actually. And the problem with this antibodies, I don't know now the cost, but at that time when we start this work, one dose of this antibody, monoclonal antibody, was 4 lakhs. If you give the 4 dose which is required to treat the cancer patient, then it will around 15 to 16 lakhs actually. And even after that, there is no guarantee that it will work or not work. So question is how to help the community to understand this part. So what we did, we collected all the information about the patient, where it works, and the patient where it doesn't work. As well as we got the information about their genomic profile. So that complete information is provided in the database. So it is important to use this information to find out the biomarkers which can judge whether this particular antibody will work for the patient or not patient based on the genomic profile of the patient. Right? Similarly, we go for the other works like oncology, cancer oncology genes, means where we work on the pancreatic cancer methylation database. So what we did actually, we collected methylation related informations, methylation status of the genes in pancreatic cancer space. So this database again provide complete thing. Please remember one thing, it's very easy to use these databases. I would like to state here that all the work, all the database, they are published actually in rapid journals. It means they are peer reviewed. It's not that we create some database and somebody will question that whether this database is valid or not valid. So we create the database, we maintain the international standards. So, and after that we publish it. So publishing, why publishing is important? When you publish in a rapid journal, peer reviewed journal, then the reviewers, international reviewers or independent reviewers will evaluate you. And based on that, it will be published. So all work, whatever I'm presenting or whatever have been developed at our group is published work. And second thing, the major emphasis why our databases and tools are popular. One of the reason is basically it's easy to use. Anybody simply can search by clicking here or browse the data on the different categories. We provide the interface, which is user-friendly interface, where the user can retrieve it. This is basically another type of the data, basically, where we have collected the epigenetic modifiers. Epigenetic modifiers. So we have different type of information, mutation data, copy number data, all type of information has been collected here, and you can use them. Similarly, it's not practically possible to study in the detail all the pathways. So we have taken the one pathway called apoptotic pathway, which is very important in the cancer. And for all cancer, we have analyzed the apoptotic pathway of all cancer patients or cancer cell lines. So this is the information provided in this database. So you can use it. This is basically ORS-TDV is a resource to explore the therapeutic potential of oncolytic virus. Though we already know, all knows that virus is responsible for the different type of disease. But same time, their number of viruses are used nowadays to do the management of the disease basically. How this is an example. There are some viruses which work as oncolytic viruses which can use to treat the cancers as well as to deliver the immunity actually. So that's a very important to collect and compile. The problem I will tell you. Sometimes I feel I'm working in this area from last 20 to 25 years actively actually. And we are always busy because there are too many problems which have not been solved at all actually. Because sometimes people say that it's very difficult to find the problem actually. And for me, I find that too many problems are there which is not solved by anybody actually. Though a lot of literature is there on oncolytic viruses, but there was no database is available on the oncolytic virus. So what we did, we collect and compile this information and we create this database. So anybody working in the field of oncolytic viruses or want to use the viruses for the treatment or vaccination purpose, then they can use these viruses for that purpose actually. This information will be utilized by the scientific community. Okay, so this is the just architecture of the database, that how many records are there, what type of articles are there. Now you will ask the question that why you create the database, because the information is already there in the literature. The problem is there. Let's say if you want to work on this one, you have here information is compiled from 5,456 research articles and 471 research articles, patents basically. It's not practically possible for anybody to read all these papers because our team work hard and then collect the data and then ultimately we get this information. So rather than everybody is searching the information, we collect, compile and provide the up-to-date information which is complete. Similarly, we go for the another database that's called cancer liver database. So this database provides the different type of biomarkers already reported in the literature. Okay, we just collected that information that may be based on the protein-based biomarkers or mRNA-based biomarkers, any type of biomarkers which is discovered in the literature last number of years have been compiled here and available there. So you can use them. There is another interesting part. There is a promoter region. In promoter regions, you have the transcription factors. Transcription factors where the transcription, and there you have the enhancers. And these enhancers control the expression of the genes. And it has been shown in the past that these enhancers, copy number, these enhancers, they directly impact the survival of the cancer patients on other records. So we compile all the information from the different sources and we put a database, cancer database. So this is available. You can use it again. So far, I talked mainly to you about the compilation of data or creation of databases. Now I am going to talk to you about more data mining or prediction. Because there are two parts in bioinformatics, I can see it. One is compilation of data, where you collect and compile the data, or you compile the data sets. That's a one business. And unfortunately, if I go back in the history, most of the Indian continent, even Asian continent, they are not creating the database. They are just using the database from NCBI, EBI, or maybe SysProt, or a number of resources. But they are not involved in the creation of the databases. So what we did actually, we take the initiative and number of databases now developed from India for this purpose. And not our group, number of other groups are also contributing in the area of database development. Because it's very important to develop the database, you got the depth information as well as community worldwide use these databases. because information is the most important part. So the problem, I will tell you, the most of the researchers who are working in the theoretical science or maybe in bioinformatics, what they are doing actually, they are just becoming a user. They use the softwares developed by somebody, they use the database developed by somebody, and they do some standard protocols, and then that's all actually. I'm not against that. That's perfectly all right. But if you want to become expert in the field of information science or bioinformatics, you should contribute fundamental. And how you do the fundamental contributions? Let's say creation of resources, creation of databases. That's a one part of the pocket. Another is you have the data. From this data, can you develop prediction methods to predict the properties of biological molecules? Or you have to do the classification, let's say normal versus cancer patient or anything. So here, data mining is important. So if you want to claim yourself that you are a bioinformatician, I will advise you to develop your own softwares, which is better than any existing softwares, and then provide to the public. Then your contribution will be there. If you are just using somebody's software, just going on the browser and searching some database softwares, don't claim then you cannot be a bioinformatician actually. You are a simply biologist, can do that job actually. So you have to rethink about your career actually, what you are doing actually. You want to become a biologist who knows a little bit bioinformatics, or you want to become a bioinformatician. If you want to become a bioinformatician, you have to think what you are contributing to the community. Are you simply a user or you are also a developer? Because number of bioinformaticians produced by this country they are simply user. They know how to use it. And that's not a difficult job at all actually. Anybody can use it. It's like that mobile. If somebody use the mobile and he start to claim that I am a computer scientist, I know how to use the mobile. That's complete nonsense. For a computer scientist, the person should know how to develop the new mobile application apps basically, or new softwares, new operating system, new thing actually, which is development part, rather than using it, anybody can do it. Anyway, so here I will cover about some important part like personalized vaccine and drugs, promiscuous cancer drugs, then stage and other biomarkers, and then some solutions for the cancers. And it is not practically possible to cover my all tools related to the cancer. I am just picking up few tools only here. If you are really interested in my groups, what type of tools are there, and you want to utilize in your research, you can visit my website. So the cancer top, that's a very interesting tool actually. We know developing vaccine for, let's say, COVID-19, we got now actually Covaxin is there, Covishield is there, Moderna, Pfizer, number of vaccines are there. There are a number of vaccines are already there for the tuberculosis like BCG and other factors. Not even. But you will realize that it's very difficult to develop a vaccine against the cancer. Why it is so? Because in case of the bacteria, virus, or any pathogen, you got the attack from the outside. So enemy is different from your own system. Like attack from the outside the country. Then you recognize that this is a Chinese attack or Pakistani attack or anybody's attack is there. You can easily kill them because you know how to discriminate between Chinese and Indians. But think about the terrorists who are basically misguided people from our own population. They are like us actually. They look like us, they are here. So it's very difficult to discriminate them, to identify them and to kill them. It's nearly impossible. That's why it's very difficult to develop the drug or vaccine against the cancer. Because if you start to kill these cancer cells, then they are similar to the normal cells. So your drug will not only kill the cancer cell, it will kill also normal cell. Now question is what to do? So here we propose the strategy. Propose the study that what we did actually from the different databases, we find out the cancer sensitive genes where the mutation is very important, because in some genes, if you mutate or not mutate, they will not convert into the cancer. But some genes, if you make the single mutation, they may become the cancer. So first we identify 60 vaccine candidates in the whole human genome, because the whole human genome have the 20-30,000 genes basically. So out of that we find out 60 vaccine candidates. These are the basically proteins which are very sensitive to the cancer. If you make the mutation, the chance is there that it will create the cancer. Then next part, what we did, we generate the 9 months, or because our immune system, MSC binders or HLA binders, they recognize at least 9 amino acids, means peptide of 9 amino acids. So we generate all possible 9 months, and then we check whether the same peptide is available in normal cell or not. If available in normal cell, remove it. If it is not, then check these peptides. These peptides are those peptides which are found in the cancer patient, but not in the normal patient. Sorry, not in the healthy person. Okay, so these are the called cancer specific peptides. Then check whether they can activate our B cells, T cells, which is required to create a vaccine basically. And the peptide which can induce our B cells and T cells, they can become the vaccine candidate. Based on that, we propose this model, cancer drug basically. And there is a two type of things, partially personalized and fully personalized. In fully personalized, what we suggest is basically take cells from the tumor and do the genome sequencing. Take the healthy cells near to the tumor, adjacent to the tumor, and sequence it. Then you have the two genome, compare it, and find out the regions which are mutated in cancer. Means that region or that peptide is not available in the healthy cells, but they are only in cancer cells. And then check whether it is a B cell epitope or T cell epitope. If it is B cell or T cell epitope, you can use this peptide or epitope as a immunotherapy or vaccine therapy. So that's a basically fully personalized, and this platform which we develop support the users to perform all these activities. Okay. So say we go for the VLC virus, a webisode for designing subunit vaccine candidate. That's another interesting part. So far, we know only about the some cancers which is created by, which is let's say generated by some viruses actually, okay, HPV and other viruses. But nowadays, research shows even there are more viruses which are not known very well, but they are also creating the cancer. So one example is lung cancer. Actually, in lung cancer, it has been shown in some cases that virus is responsible for that, and we normally find out the viruses which are responsible for the lung cancer actually, and then we develop the subunit vaccines, means epitope based vaccines, means possibly suggested subunit vaccine, because that's easy to handle actually, because it is generated created or by the virus actually, and virus is different than the normal healthy cells. So it's very easy, means it's easy to find out the vaccine candidates. So we propose in this one what type of vaccine candidate will be there. So anybody is working in this area can use them actually. There is another important platform we have, that's called prioritization of anti-cancer drugs. Please remember one thing. It's not practically possible that you take the genome of a cancer patient based on the mutations, you develop a new drug, because drug development is a very long process. It takes years and years. In the meantime, the patient will die actually. So that's not practically possible to propose that type of strategy. So what we propose, that already in market, number of FDA approved anti-cancer drugs is there. Question is, which drug should be tried, or which drug will be suited most to a given patient based on its genomic profile. So in order to address this question, we have developed this platform. If you give the genomic profile or expression or whatever, then it will predict, or it will prioritize or rank the existing cancer drugs, that which drug will be suit best to this particular patient actually. Okay, so that's why we call the prioritization of anti-cancer drugs based on the genomic profile of the patient. Okay, so that's the title of this paper: prioritization of anti-cancer drugs against a cancer using genomic feature of cancer. Okay, so if you have the genomic profile of the cancer patient, then it can help you the priority of the different type of drugs which is available in the market. Okay, and we have demonstrated that all drugs are not suitable, because here are the name of the different drugs, and these are the name of the different cancer type of cells actually, cancer type of cancers actually, bone cancer, breast cancer, skin cancer, and the color itself so that how effective it is. And this is basically performance of the different drugs. We have developed the prioritization for different drugs based on the different features like mutations, variations, expressions, copy number variations, and this is the hybrid model which combine more than one property. And we demonstrated that predicted and actual IC50 value is have the 0.78, because 1 is a maximum. So it's not perfect, but it's quite good actually. This is another method, because what we are doing from last number of years, something different than the normal public is doing. Normally, we take a cancer, we develop the inhibitors based on the inhibitors on the target. Okay, but there are problems. Even if you take the pancreatic cancer, I'm giving you one example of pancreatic. There is not a single type of pancreatic cancer. There are different type of pancreatic cancers are there. So if you want to design a drug, the drug should be in such a way that it works with the all type of variants of pancreatic cancer. Okay, so all subtype of cancer should be treated by this drug. So here the attempt has been made where we call the promiscuous anti-cancer drugs. Okay, so these are the similar things. I'm not going in the detail of each and every one. So that's another idea of platform independent biomarkers for this hepatocellular carcinoma, liver cancer, and why we are giving the important identification of platform independent means there are different platforms for measuring the gene expression. It may be Affymetrix based microarray. It may be cDNA microarray. It may be RNA-seq based. The question is, can we develop a method which work for any data which is coming from the different type of platforms? As well, here we have used large-scale transcriptomic data actually. So ideally, your biomarker should work in such a way that it work on any type of data basically. Similarly, there is another method for classification of early and late stage of the cancer. That this tool is there. You can use them actually. Cancer TSP. I'm just jumping them because they are repetitive things. Only thing, if you are interested, you can use them. This is for skin cancer progress. That whether it's a localized metastasis or global metastasis, or it go beyond the nodes, it based on the genomic profile, we propose a method to predict it. Okay. This is based on the skin cancer based on the HLA typing, because you know immune system is not same for all the persons. Actually, we have the different immune system. Can we predict that based on the immune system, if the person is suffering from cancer, how risky it is? Because in some time that if you have the HLA typing, proper HLA typing, then the particular cancer will not survive for long, or it will be the long life. The person will survive for long life because they have the protection. But if HLA typing is missing, the particular type HLA is missing, then it's very difficult. So here, based on the HLA typing, we suggest the prognostic biomarkers. This is based on the protein-based biomarkers. So we have used a different type of biomarkers, expression-based marker, HLA typing-based biomarkers. Okay. So this is the way how you will predict it. You will give the protein concentration. It will predict that what is the risk actually. So this is again for diagnostics in body fluids, the database. And this is now question is what type of solutions we are providing, because we are not in the pharma business, we are in the biological base. So what we are suggesting is biomolecules based solutions against the cancer. Means the molecules which is generated, which are natural molecules. Let's say, what are the natural molecules in your living organism? You have the RNA, you have the DNA, you have the protein, and you know protein or peptide, they are heavily used in your living organism. Can we use protein or peptide as a therapeutics? That's a challenge. So what we propose here, that how to predict the anti-cancer peptides. So this database, this server allow you to predict whether your peptide is anti-cancer peptide or not. Or you want to design the better anti-cancer peptide. With the help of this software, you can design the better anti-cancer peptides. So this is a recent method we have already developed recently and available to you. This is for drug delivery in the tumor. The one of the problem is how to deliver the drug at the site of the tumor, because it will go to the other places and will not reach on the tumor site basically. These peptides have the property that they, if you conjugate any drug, they will specifically go to the tumor site. So they have the affinity, these peptides have the affinity with the tumor. And we collected and compiled this database, so you can use it. As well as prediction method is there, you can predict whether the particular peptide is tumor homing or not. Actually, if you are really interested in this area, I will advise you to read this review. This is called Computational Resources for Identification of Cancer Biomarkers. This is published this year itself actually, and that's part of the editor choice. They make it free available to the public so that more and more public can use it actually. And it is published in Briefing Function Genomics. It's a good journal. So I will advise you that read this paper. This is freely available. You can read it and you will get all the information, not from our group only, from the other groups as well. So that's the advantage for this review, that it will cover most of it. So thank you very much. That's all for today.
Thank you, Singh, for your nice talk. Sir, your lecture was highly inspiring, and you have given a lots of messages to the young faculty members, scientists, and researchers. How can they use these web servers, tools, and methodologies in their studies? Sir, since last many years, we're actually following you, reading your articles, and today we're really blessed, sir, to get you at our institute. Sir, with your permission, can I take a few questions from the participants?
Yeah.
Okay. Sir, Dr. Ramachandran, he's, I think, very happy with your lecture. He's mentioning very useful information, and he's asking, do we have any databases for melanoma cancer in India?
I don't know that whether specific to the melanoma cancer, there are a lot of databases are there actually. You can get it. For specifically for the India, that's I don't think so. And I have the suggestion actually. Because the problem I have seen in the most of the researchers in Indian community, they are talking about India, India, India. Actually, really, if you want to become a good scientist, you should forget about the boundaries. Let's say if you want to create the database, create for the world. That should include the India also. Actually, creating
Right,
for
sir.
the only small fraction will not give you the recognition in the world. Actually, that's my suggestion to the young community. Because I have seen a lot of people, they are talking about India, India, India. Because when we talk about the research, we are talking about the scientific community. We are talking without any boundaries of anything. So if you go to the melanoma cancer databases, there are number of, okay, you will get the information. Only
Right, sir.
problem there, I will tell you another problem, which because these are the important point. New generation should understand this part. Unfortunately, we want to use others' data. We are very happy to use the others' data, but we are not good in sharing our own data. And I have seen in number of agencies in DBT, DST, we are forcing number of times PI, please submit your data, but they don't want to submit it actually. They want to die with the data, but they don't want to share with the data public actually, because they feel they will live forever actually. They will never die, and what will happen later on? They will die, and the data will die. So please change this attitude. That's the most important part. We are as intelligent anybody in the world actually. I have worked in the number of countries. I don't find any lack of talent in this country. Only thing, lack of attitude towards the science and other things actually. We are become so selfish actually. We think about ourselves actually. And in this business, we are not helping anybody or not helping ourselves actually. These are some suggestions to my young colleagues actually.
Right, sir. Thank you, sir, for enlightening us. Sir, I think Shurajit Pathak, one of our participants, he's also so much agree with you, and he's also mentioning, sir, what about the validation of Indian databases? Very few follow up to date.
No, question is basically Indian database. Again, the question is basically whether developed by Indians or developed for Indian need actually. These
Right,
are
sir.
two different things actually. Like if you talk about the databases developed from India, if we see the overall average in the world, maintenance or updation is a major challenge. Not for India, all over the world, because one study shows in next 5 years, last 5 years, 50% databases or prediction server shut down actually. They are not able to maintain it. That's a major problem worldwide actually. So overall statistics is same India as well as for abroad actually.
Great, sir. You have very nice, rightly mentioned that India is led, and sir, because of great scientists like you, our country will rise. And in your website, where you have shared that A to Z web server lists, sir, we're very happy, and this is really a great inspiration for us, sir. And our young generation, we hope we will try to follow this path. Sir, I have one question from my side. Sir, since you lecture in drug discovery hackathon, and during sir lockdown period also, you have given a lots of lectures. Sir, you have mentioned and you have highly popularized about the peptide based therapy. So sir, what will be your suggestion for us? Because sir, like for example, faculties like me and other participants, all of us are from pharmacy background. We are actually too much dependent on small molecule based therapeutics. So sir, what will be your advice for us? How we can go through with the peptide based therapy, and what should be our approach, sir?
Okay. I will tell you very interesting part. I hope time is there. Time is not a
Yes, sir.
problem.
Yes, sir. Yes, sir. Please, sir.
I will tell you a very interesting story. Sometimes our seniors confuse more than providing the correct information. Let's say I will tell you that 7 or 8 years back, I talked to the chemist and I asked the same question whether we should to go towards the peptide based therapeutics or we should to focus on the chemical based actually. So what the person say no, no, this is a fancy area actually. Protein and peptide based drug is not feasible practically actually, and he criticized lot. I keep quiet, okay, because if you don't have the knowledge, you cannot argue it. What we did actually, we create a database, and that database is already there, THPDB,
Yes, sir.
therapeutic protein and peptide database, THPDB. And what we did actually, from FDA approved, what we did, we collect and compiled the FDA approved drugs, and we created a database. And that database itself, so it was in 2017, that it shows that nowadays FDA, which is basically international USA based agency, is now uh, approving more and more peptide or biologic based drugs rather than small chemical based drugs actually. That's the change actually in last 2 decades actually. Now, number of drugs approved every year. In case of chemical, it is static or not changing, but in case of peptide and protein, it is increasing. So it's a time in the future that more and more biologics based drugs will be there. Why it is there? Because toxicity is one of the major problems. And for these chemicals, we don't know what is the long-term toxicity. Okay, but the protein or peptide or biologics based, the one advantage is there that they are from the natural system. So they will automatically degrade it over the time with the enzymes we have in our body, because our body itself handling this type of things. I'm not saying the challenge is not there in the peptide protein. There are other challenges. So I will advise you that start this business also. This is going to come in the future actually. That's what I feel actually. And this THP database in last 2 or 3 years, it got more than 250 citations. So now you can understand worldwide how many people are reading our database and using it actually. So heavy demand actually. That's all actually.
Right, sir. Thank you, sir, for showing us the path. So I think our honorable, the convener of this FDP, Dr. Pamajundar ma'am is also here. Ma'am, do you like to add any?
Thank you so much, sir, for throwing so much light on the various aspects of computer-aided solution for managing treatment of cancer. Sir, thank you for accepting our invitation and giving your valuable time here. And we are very sorry for the beginning part because there were some technical problems from our side. Sir, hope you will be accepting our invitations in future programs also. Thank you, sir.
Okay, yeah. Thank you everybody. Bye.
Yes.
Thank you, sir. Have a nice day, sir. Okay, it was a great time with Dr. GPS Raghava sir. And my dear participants, today it is a holiday, but despite the holiday, all of you are here for learning, and it's a great encouragement for us.